Nolan was able to send me his stuff first, which was great. Shortly followed after, Austin was also able to send me his PowerPoint. And then lastly, Josh sent me his through Facebook, which was really convenient to get everything nice and quick on Saturday. Section 3.11 deals with proteins. Proteins are made up of amino acids, and then amino acids are also made up of two different groups, the hydroxyl group and the carbonyl group. There's a third group involved that's known as the R group, and the R group is sort of like the wild card. It can be many different things. The R group is just a side chain specific to each amino acid. The R group is really important because it is the functional group attached to the amino acid. There are two types of amino acids. There are those that are hydrophobic, and they fear the water, and they will repel the water. Then there is also hydrophilic, which will bond with water, and that's really just determined upon charge and polarity. How are amino acids connected? It doesn't require calculus to figure out this. It's one of two reactions, both of which are fairly simple. The dehydration reaction, which takes away water, will combine carboxyl groups and amino acids to hydroxyl groups of another amino acids. The covalent link is known as a peptide bond. The other type of reaction is a hydrolysis reaction, which requires the adding of water and can break peptide bonds. The shape of a protein determines its function. There are 7 major types of proteins. There are structural, contractile, defensive, signal, receptor, transport, and storage. Most enzymes tend to have a globular shape, while structural proteins are thinner and are called fibrous proteins. The shape of a protein plays a large role in its function because it needs to bond to other molecules to do its job. Once a protein's shape is altered, it is in a process called denaturation, and it loses its function. This can occur from a change in the salt concentration, a change in pH, or heat. There are 4 levels of structure within a protein. The first requires amino acids to be in the correct order. Another major point about the first level is that any minor changes to the first level will have major ramifications further down the levels of structure. The secondary structure is where polypeptide coils fold into patterns. The coiling pattern is known as the alpha helix. The folding pattern is known as beta pleated sheets. Both are spaced by hydrogen bonds and oxygen atoms along the backbone of the polypeptide chain. The three-dimensional shape of a polypeptide determines the function of a protein. The R group helps determine the shape, and the protein shape may be further reinforced by covalent bonds called disulfide bridges. The final level is called quaternary structure. Subunits are the polypeptides that associate with each other that make up the quaternary structure. If there are slight changes in this level, diseases such as Alzheimer's or Parkinson's can occur. This enforces the importance of the unique shape of the protein as it determines its proper functioning.